In Chapter 5, we will discuss extensions and modifications of basic principles of inheritance postulated by Gregor Mendel. Although his principles of inheritance are still valid, they are not sufficient, or they cannot explain the inheritance of all genetic characteristics. There are many traits. When you examine those traits, you will see that there are additional factors which affect phenotype of the offspring, and these factors alter the phenotypic ratios predicted by Mendel's principles, and we will discuss these factors in this chapter. Beside formulating law of segregation and independent assortment, Mendel also formulated a concept of dominance. Nowadays we know, if we translate his observation and his concept, we know that each gene comes in two versions, respectively each gene has two alleles. And what Mendel formulated, if we translate his formulation, we come with concept of complete dominance. So in complete dominance, basically one allele masks appearance of another allele. So when you look at heterozygous, you will see that phenotype of heterozygous is the same as phenotype of homozygous dominant, so recessive allele was masked. This concept was very important and applied to crosses which Mendel performed with garden peas. However, not all traits follow complete dominance. We have genes, allele of which fall under incomplete dominance or codominance. So how you actually define whether you look at complete, incomplete dominance or codominance? You always look at heterozygous. If phenotype of heterozygous resemble dominant homozygous dominant parent, we look at complete dominance, and phenotypic ratio in F2 generation will be 3:1 because two heterozygous and homozygous dominant would show the same phenotype. In incomplete dominance, heterozygous is phenotypically in between of two parents. It does not resemble neither of the parents. So you have one of the nice example in book, which is eggplants. In eggplants, homozygous dominant plant is purple. Heterozygous is violet and homozygous recessive is white, so that violet fall in between of the two. So what is the outcome of this? In F2 generation, what you would observe, let's go back to eggplants. You will see one purple, two violet, and one white. So you will see phenotype in the ratio 1:2:1. So in incomplete dominance, phenotypic ratio of F2 generation equals to genotypic ratio, and it's because the heterozygous falls phenotypically in between of two homozygous. In codominance, there is no dominant or recessive allele. The allele are codominant. What does it mean? That heterozygous would express both allele. So basically, when we look at the dominance, and what we should emphasize here is that dominance is a result of interaction of alleles of the same gene, allele on the same locus. So dominance is actually allelic interactions, and dominance is therefore interaction between the products of the genes. So keep in mind, dominance equals allelic interaction, one gene, one locus. Predicting outcome of genetic crosses, we always consider that if an individual possesses or has certain genotype, the genotype is also manifested phenotypically. phenotypically. However, this does not happen all the time. There are certain genes which do not have complete penetrance. What does it mean? It means that certain individual may possess a genotype for certain phenotype, however, he or she does not express it. So what is penetrance? Penetrance reflects the percentage of individuals who have a particular genotype, and this genotype is expressed in phenotype. Why do we consider this? Because it is important in predicting outcome of genetic process, because incomplete penetrance alter phenotypic ratios which you look at. Let's consider that allele A of gene A is dominant, however it has only 50% penetrance. So what is the outcome of this? How do you take it into account? So let's say if you cross homozygous dominant with homozygous recessive, what you expect in F1 generation? You expect that all offspring being heterozygous would manifest the trait A, dominant allele, because we consider simple dominance. However, if the penetrance is only 50%, it means that only 50% of those individuals who have this genotype would also manifest phenotype. So it means only one half would be phenotypically dominant and one half would be phenotypically recessive. If the penetrance was 30%, only 1/3 will have the phenotype. So in such case, we talk this incomplete penetrance actually alters the expected phenotypic ratio. Another phenomenon which can actually alter the phenotypic ratios is expressivity, which is the degree to which a characteristic is expressed. Let's consider human polydactylia. There are many different forms, but one of them is encoded by autosomal dominant allele, and it's highly prevalent in Amish population in the United States. In this case, the people who do have the gene allele for polydactylia should theoretically express it. However, not all of them express it. Only portion of people, individuals who have gene for polydactylia, meaning having 6 fingers, do express it. So it has incomplete penetrance. Also, it can have variable expressivity. So individual who has genotype for polydactyly for 6 fingers, that person can either express full functional finger, so that is full expressivity, or it can only manifest partial finger, or it can manifest only skin extension, so it can express it to different level, so there is variable expressivity. So why does this happen? What is the reason for incomplete penetrance and variable expressivity? That might be several reasons. It might be effect of different genes which alter expression of a particular gene or alleles. It can be due to environmental factors that alter, which suppress the expression, or sometimes environmental may enhance expression of particular allele and gene. Another phenomenon that can contribute to the alteration of expected phenotypic ratio is appearance of lethal alleles. These alleles cause death at an early stage of development. So basically the genotypic constitution which causes death, that phenotype encoded by this genotype would not appear. You have some example in book from yellow leaves of the snapdragon and the mice. So basically in mice, for example, yellow color is always heterozygous.
Homozygous recessive is white, but you never have homozygous dominant because it's homozygous allele Y for yellow encodes color. But when it's in homozygous constitution as a homozygous dominant, it acts as a recessive lethal allele. So basically, Y recessive because you need two Y allele in order to cause death. So it does act as a lethal allele.
Most of lethal alleles are recessive, which means that you need to have two allele in order to manifest lethality. There can be also lethal allele which are dominant. However, truly dominant lethal alleles cannot be transmitted if they manifest during embryonic development, because every individual who will have at least one of this allele would die before birth. Only they have dominant lethal allele can be transmitted as if they have delayed expression later on after the birth. In summary, most of the lethal alleles are recessive in nature. Most of the genetic system that we have examined so far had only two alleles for one gene, one locus. However, there are traits, there are genes in which the locus for that particular gene can select for multiple alleles, and we refer to it as a loci with multiple alleles. One of the best example is MHC class 1 and 2 system. Now, keep in mind also, loci can select from many different alleles. The genotype was an individual genotype. Each individual can have only two alleles. So what does it mean? When there is locus with multiple alleles, so basically from this multiple allele, two are selected, so more alleles there are, higher variability it brings. Let's say if you have a system with 5 different alleles, so number of alleles is 5. Now we know that in one gene, in one locus, we can have only 2 alleles. So how many possible combination, how many possible genotype we can have? So for this you use a formula where n is multiplied by n plus 1 and divided by 2. N equals number of alleles. So if one locus can select from 5 different alleles, so number of possible genotypes are 5 multiplied by 6 divided by 2, which is 15. So there are 15 possible combinations. So you can see how this increase variability, because instead of one genotype you can have 15 different genotypes. So blood group ABO belong to the system with multiple alleles. There are 3 different alleles. There is allele IA, which is dominant to allele lowercase a. There is allele IB, which is also dominant to recessive I, and allele IA is equal to allele IB. Equal, what I mean by that? They are co-dominant. If these two alleles are present, both are manifested. So this one will be blood group A, this will be blood group B, and this one will be blood group AB. So blood group O is determined by homozygous recessive. Now, how blood groups are formed? If you have a cell, red blood cell, okay, right here, in red blood cell, there is a blood groups, ABO blood groups are formed first by the carbohydrate moieties, which is attached into the glycolipids or glycoprotein on the surface of the cells. And this glycolipids or glycoproteins has this terminal strand of sugars here, where the fucose is important docking sugar. Now, once you have this chain, basically, if there is nothing added to it, this basic chain does not trigger antigenicity, does not act as an antigen, so does not trigger immune response. What triggers immune response is this terminal sugar which is added to it, and it can be either acetylgalactosamine if individual possess enzyme which adds this terminal sugar and acetylgalactosamine, it would be A, allele A, or blood group A determination. If that person has enzyme which has galactose, it would determine allele B or blood group B, and this person who has both enzyme would have blood group A and B. Now, if there is no enzyme, as here, so there is no the terminal sugar, the person in this case would be homozygous recessive and have blood group O. How and why it is important? Blood groups such as ABO are very important in transfusion. I said such, ABO and RH, rhesus factor. There are other blood groups. There are over 30 different antigenic blood groups on the surface of red blood cells. However, from all of this, only ABO and RH are antigenic, meaning they are only two blood groups which trigger immune responses. Other ones, such as MN blood group, which by the way is also codominant because it's encoded by locus, which can have allele M or allele N, and it's MN blood group, but this blood group does not trigger any immune responses. So it's why we only type and assess ABO and RH factor, because those are very important to assure compatibility between donor and recipient during blood transfusion. So how does it work? If somebody is blood group A, this person has genotypically or can be genotypically either homozygous dominant or heterozygous. And it does in both cases express antigen A. So this antigen A is expressed on his or her own cell, so immune system recognizes it as a own and it would not attack it. However, if this person gets transfusion of blood group B, the immune system does not recognize B as a self, so it would form antibodies and it would trigger coagulation right here. So when you took person blood from per individual with blood group A, this person can donate blood only to person of the same blood group or AB, because AB has both A and B, so it does recognize this A as own. The same apply for B. B can be encoded either as homozygous or heterozygous, and it does trigger formation of antibody if encounter blood group A, because A is not self. So person with blood group B can donate blood only to B and to AB. AB actually can have only one genotype because alleles A and B are codominant, both are expressed. So this person recognizes both antigens A and B as a self-own, so it does not produce any antibodies. However, it has both antigens, so it can only give blood to AB. Now, blood group O, which does not possess any antigen, any of those terminal antigen, neither acetylgalactosamine or galactose, so it does not have any antigen. So basically, both antigen A and B are recognized as a foreign, and person with blood group O would form antibodies against both of them. So basically, a person with blood group O can only receive a transfusion from person with blood group O. However, since he or she doesn't have any antigen on surface of its red blood cells, that person can give blood to every other blood group, and it's why they are called universal donors. In contrary, did you notice blood group AB is universal recipient? Now let's now talk about gene interactions. We have discussed previously dihybrid crosses in which two independent genes actually their phenotypes are combined in dihybrids and yields a ratio 9 to 3 to 3 to 1. Let's consider round and yellow seed. Round and yellow, which alternative can be green and wrinkle. So these two genes, one for color of seed and one of shape of the seed, are two genes which actually we follow how do they assort, but final phenotype reflects this gene independently. What does it mean? You can have round and yellow, round and green, wrinkle and yellow, wrinkle green. So one gene does not affect expression or phenotype of other gene. However, since many of the characteristics are encoded in the metabolic pathways where one intermediate is actually substrate for another, and one conversion is catalyzed by one enzyme, second by conversion by second enzyme, and each enzyme is encoded by different gene. Under such scenario, basically one gene affects appearance of second one, and we talk in this case about gene interactions, and they can be simple gene interaction when two gene actually forms one phenotype. We will talk about let's say pepper, color of pepper, or they might be in epistatic relationship where one gene masks the effect of another gene. Now, one masks other. You recall dominance and recessivity. Be careful, because in dominance, one allele masks appearance of another allele. In epistasis, one gene masks appearance of another gene. Let's first examine very simple gene interaction in which two genes interacts and produce one characteristic or one phenotype. You have an example of the fruit of pepper, which can be either red, peach, orange, and cream. So how this color is formed? The final color is a result from the relative amounts of red and yellow carotenoids. Each of them is synthesized in complex biochemical pathway, and each pathway is actually regulated by different enzymes. So as you can see here, you have two genes, two loci, gene Y, and gene Y, let's say, encodes enzyme which works in first pathway, and gene C encodes enzyme which acts in secondary biochemical pathways. So if you have at least one dominant gene allele in both genes, in each of these genes, basically you synthesize maximum amount of the carotenoids, red carotenoids, and basically the color would be red. If there is a dominant allele in gene Y, but C gene has both allele recessive, this will yield peach color. If Y is recessive, homozygous recessive, and C gene has at least one dominant allele, you will get orange or orange color. However, if both are recessive, the color will be white. So basically, this is interaction of the accumulation of the carotenoids, and basically they are synthesized in complex pathway where different enzymes contribute to final amount of each color. So they do interact. What does it mean? If you have normal dizygote, if you have two genes where genes do not interact, you get ratio 9 to 3 to 3 to 1. Let's say you will have, if we go back and talk about the round and yellow seed, you will have 9 plants with round and seed with yellow color. You will have 3 round with green color, 3 wrinkles with yellow color, and 1 wrinkle with green. So you will see a distribution of both phenotypes. If gene interacts, in this case, you also get ratios 9 to 3 to 3 to 1, because in both cases there is segregation. However, each one only creates one phenotype: red, peach, orange, and cream. So these two genes interact to create one phenotype. So whenever you solve problems with gene where there is interaction, and you use probabilities and branch diagrams, do not plug in phenotypes. Do work with genotype, because phenotype cannot be determined without considering the effect of genotype at all of the loci which contributes to it. So keep in mind, 9 to 3 to 3 to 1 now does not reflect phenotype of two independent genes, but since this gene interacts and affect each other, this ratio actually corresponds to the phenotype, only one phenotype, either red, peach, orange, or cream. Epistasis is more complex interaction. It is an example of much more complex interaction between two genes. In epistasis, one gene masks appearance of the other one. Now, stop and think about dominance, epistasis, dominance, allelic interaction, epistasis, interaction between genes. Epistasis can be either recessive dominant or duplicate recessive. So we will consider first recessive epistasis, and we will go over 2 examples. Firstly, we will talk about the color in Labrador Retriever, and afterwards we will discuss ABO antigens in blood groups. So before we go to the example, I want you to think about how genes are manifested, how they are expressed. If you understand what is needed or necessary for manifestation of certain gene, meaning the traits, appearance, it would help you to understand epistasis. So let's go to the Labrador Retriever. Coat color in labs is controlled by 2 genes. One right here is gene for color, where black is dominant over brown. What does this mean? This gene controls synthesis of enzyme, which is actually in charge, or which controls synthesis of pigment. Pigment is synthesized in skin. Now this pigment, upon synthesis, has to be transported into fur in order for dog manifest color, fur color. And this transport is controlled by transporter, which is encoded by different gene E. So if you have a dog and that dog has black fur, like this one here. So this dog firstly has to have at least one dominant allele in gene B, which would synthesize black color. So it can be either homozygous dominant or it can be heterozygous. However, this is not sufficient. This dog has to express at least one dominant allele for gene E, because if the pigment is synthesized in skin but it cannot be deposited into fur, the final color will not be black but yellow. So if, for example, you have dominant gene B, however, there is homozygous recessive, this dog is homozygous recessive for E, what does it mean? That he does not produce any transporter. The outcome, the final color will be not black but yellow. So gene E is epistatic because it controls appearance of gene or manifestation of genes B. So gene B is hypostatic. Another example of recessive epistasis is actually gene H, which control expression of ABO. Let's step back and remember how the blood groups are encoded. You recall that on the surface of red blood cells, you have the basic chain, which is intermediate chain formed by glycolipids or glycoproteins. Now, this chain alone is not sufficient. It doesn't give a base for enzyme encoding that addition of the terminal sugar to be added to it. You remember, only this terminal sugar is antigenic, and if this terminal sugar is N-acetyl galactosamine, that person would have blood group A. If it is galactose, it would encode for antigen B. Now, in order to attach this terminal sugar, the enzyme which does it needs to have this base to which it can attach it. And this base is formed by addition of fucose to this intermediate chain. This addition is controlled by enzyme which is encoded by gene H. Once the fucose is added, it creates the H chain, which forms a base for enzymes to add the terminal sugar. If person has that enzyme, if he doesn't have, he would be blood group O. Now, what can happen rarely is that this locus H has actually two recessive allele. If this happens, there is no enzyme that would attach the fucose and the chain stays in the intermediate form. So it does not create a base for enzyme to attach the terminal sugar. So in this case, a person who is homozygous recessive for H can have alleles that would encode, let's say, blood group AB. However, it has homozygous recessive alleles in H locus. Also, it does encode enzyme for attachment of terminal sugar for A and B. It would have blood group O because those enzymes cannot function. Now, this syndrome is real. It's called Bombay syndrome. It's very rare, but it can happen. And in this Bombay syndrome, the H gene is epistatic to gene A, B, encoding either enzyme attaching blood group A or antigen A or antigen B. So basically A and B are hypostatic. In recessive epistasis, the presence of two recessive alleles is required, is necessary in order for epistatic gene to mask appearance of hypostatic gene. So it's why it is recessive, because you need two alleles. In dominant epistasis, one dominant allele, presence of one allele is sufficient to suppress or to mask appearance of second gene. Now let's look at the example of summer squash. Summer squash comes in three colors: white, green, or yellow. Now, this color synthesis of the pigment is actually controlled by biochemical pathways where two enzymes are active. Enzyme 1 converts compound A, enzyme 1 converts compound A into compound B, and compound B already gives a green color. However, compound B is also intermediate, which can be converted by enzyme 2 to compound C, which actually gives squash yellow color. Now, let's make a scenario that enzyme 1 is constitutively active, however, it's controlled by gene W. So, gene W, imagine, encodes an inhibitor. So if there is homozygous recessive WW, there is no inhibitor, enzyme 1 is active and converts compound A into compound B. However, if there is at least one dominant allele in W, so now there is an inhibitor which would inhibit the activity of enzyme 1, there would be no conversion of compound A into compound B, so all squash will be white. So every plant which would have at least one dominant allele, W, will be white. Now, in other scenario, what can happen is that there is no dominant allele, no dominant W, so there is no inhibition of enzyme 1. So enzyme 1 converts A to B. Now, not only this gives squash green color, however, compound B is actually intermediate. It is a substrate for enzyme 2, which can convert compound B to compound C, and compound C gives yellow color. However, this enzyme, enzyme 2, is not constitutively active, so you need to have at least one dominant allele which would encode this enzyme, production of this enzyme, in order to have conversion from B to C, from green to yellow. So what does it mean? That outcome of this epistasis is that every plant which has at least one dominant W will be white. Those which have recessive W, homozygous recessive, those plants which are homozygous recessive for W, so there is no inhibitors, can be either yellow or green. They will be green if there is no presence of enzyme 2, which would convert B, compound B, into C. They would be yellow if there is enzyme 2. It means if there is at least one dominant allele which would encode enzyme 2, so the compound B would convert it to C and squash will be yellow. So this epistasis also will alter the phenotypic ratio of the final product. Finally, let's consider duplicate recessive epistasis. In this case, homozygous recessive constitution in either gene 1 or gene 2 can result in no phenotype in epistasis, interference with appearance of phenotype. An example here is albinism in snails. Albinism is caused by lack of pigment, and pigment is produced in multi-step biochemical pathways. So albinism can result in the lack of the enzyme 1 or enzyme 2. So in order to get color, and dominant allele A is required to be present in gene A, which actually controls synthesis of enzyme 1. Enzyme 1 converts compound H to compound B, which is an intermediate and substrate for enzyme 2. However, compound B does not produce any pigment, so snail is still colorless. Now, a dominant allele in the locus B is required, therefore, to produce enzyme 2, and only if enzyme 2 is present, it does convert compound B, which is intermediate, into final compound C, which actually produces a pigment. So albinism can actually arise either from absence of allele A, or dominant allele A, or from absence of dominant allele B. So either snail is homozygous recessive for A or it is homozygous recessive for B. In both cases, the snail will be albino. As discussed before, gene interactions will alter expected phenotypic ratios predicted by Mendelian inheritance. However, keep in mind, what is altered is phenotypic ratios. However, Mendel's law of segregation and independent assortment still apply. What is difference? The only difference is how the products of the genotypes interact to produce the phenotype. So if there is gene interaction, you cannot consider the expression of individual genes on separate loci. You have to consider how these genes at different loci interact. Therefore, I don't want you to just memorize this table with altered ratios. What I want you is to think and to consider how actually this phenotype might be created. What does it mean? How these two genes might interact in order to produce these genotypic, respectively phenotypic ratios. Finally, the last slide of this first part recording for chapter 5 is discussing complementation. So I would like to actually stress what complementation is. Complementation is a test in which you can determine whether different mutations are a result of allele located on the same locus or whether the mutation come from different genes in different locus. So let's say in fruit flies wild type eyes have red color. However, if you work with fruit flies, you know that you can see flies with white eyes and also apricot eyes. Both of these mutations are X-linked recessive. Now, question you want to ask is, do these two phenotypes by an apricot result from two different mutations of allele of the same gene of the same locus, or are these two different genes which control these two mutations? So how do you approach it? You do complementation test. So you select two parents, one which is homozygous recessive for one mutation, let's say white eye, and another which is homozygous recessive for another mutation, apricot eyes, and you cross them. Now, if these two mutations are results of the two different mutations of alleles of the same gene on the same locus, so if they are allelic mutations, what would happen if you cross these two homozygous recessive mutants? You will get offspring which will get mutated allele A from one parent and mutated allele B from other parent. So it will have mutant phenotype, mutated phenotype. However, if these two mutations occur in two different genes which are in two different loci, and you cross homozygous recessive for A with homozygous recessive for B, if there are two loci for A and B, homozygous recessive for A would have wild type B gene and homozygous recessive for B would have wild type A genes. So basically in this case, offspring inherit from one parent recessive A but wild type B, and from another parent recessive B, mutated B, but wild type A. So basically these two different loci with two different set of alleles would complement each other and would produce wild type phenotype.